Assalamualaikum guys, we are live. We will be starting in about approximately 2 minutes. If you just bear with us, 2 minutes. Assalamualaikum guys, for everyone that is watching the live, we'll be starting in just in about a minute. Okay. Alhamdulillah Rabbil Alameen wa salatu wa salam ala Rasulullah sallallahu alayhi wa sallam amma ba'd. Assalamu alaikum wa rahmatullahi wa barakatuhu. Jazakallah khair for everyone who is attending today's webinar around the COVID-19 vaccine in particular. We're going to be speaking about the process around what is a phase 1, phase 2, or phase 3 trial. What does mRNA mean? What do they mean when they say the red tape was lifted? Was this vaccine rushed? This is a follow-on from our last webinar, which involved the 3 doctors, which is really, really good. We had some absolutely great feedback. There was some who were asking that, some of the questions weren't answered, or it wasn't in depth. Our guest today, Qamar Hussain, who is a senior scientist who works for Glaxosmithkline, very kindly came forward and said that he could go a bit more in depth with some of these topics. So we thought, yeah, that's great. We could get through some of these topics and hopefully dispel myths again. Again, another disclaimer, the same as last week or a few weeks ago. We aren't saying that, yes, you should take the vaccine or no, that you shouldn't take the vaccine. It's just, if you make a conscious decision, it should be at least based on the correct facts, not on what someone has forwarded in a WhatsApp group or someone's just tried to copy and paste. Or in fact, it's also the way research is done. Sometimes you have a lot of scaremongering, sometimes you have a lot of articles that come forward and you're not really sure how to verify them. So for example, in Islam, we know that if you want to verify something, you go to the right sheikh in the right area, whether it's Usul al-Fiqh or Ulum al-Hadith or something like that. Here, inshallah, we have someone who can also research on top of what he knows from his past 5 years of working. So I'm just going to pass it over to Qamar, who's just going to explain what he does and how he will hopefully tackle these questions that we're about to start. Qamar.
Assalamu alaikum. Jazakallah khair, Hassan, and the brothers for having me on this webinar. Just want to kindly introduce myself. So I did and I'll talk a bit briefly about my background. So I did a bachelor's and a master's degree in biomedical material science and tissue engineering. So these degrees are covered a lot around material science. So looking at different types of materials for biomedical applications. So metals, metallurgy, polymers, ceramics, composites, nanomaterials, going right down into the subatomic level to look at how these materials undergo different type of stresses. The other half of the course was the biology part, so covered areas of biochemistry, protein, biochemistry, a lot around stem cells, so stem cell science. So when you merge the two together, it's all about the interface between material science and human biology and how we can combine the two to make materials for biomedical applications. So really, a simple example is in your mouth, you have fillings. So you have to choose the right filling you have to choose the right material to use for that filling, because every day you're chewing on food, so that material has to undergo certain types of stresses. Another material is, for example, contact lenses, heart pacemakers, hip prosthetics. So it's very in depth. But the course would mainly focus around regenerative medicine. So using stem cells to potentially grow organs and replace people with kidney diseases, or one of the biggest ones is how to grow a human heart, a whole human heart.
Wow, okay.
If we're not quite there yet, but I've been to a conference.
So what do you do in your day job?
So alhamdulillah, after 2016, I managed to get myself a job at GSK. I've been there for 5 years now. So I work in the department of cell and gene therapy, and in that department, I work in the discovery research side. Cell and gene therapy is quite a broad area of science, which has taken off over the past 10 years. More so, over the past 5 years, it's taken off quite a lot, and it's changing the way we treat diseases and cure diseases. There's a lot of potential to cure some diseases in the next 2 decades. You can cure blindness. Maybe there is the potential to cure diabetes, and also cancer. And we are actually at the stage where there's actually 4 products on the market that are curing people with leukemia. Completely come out into remission with no relapse. So the cell and gene therapy area is a lot of it's to do with gene editing as well. The area that I'm involved in is cell therapy and gene therapy for immuno-oncology. What does that mean? That means cancer immunology. So cancer obviously is a big disease, and it's probably the disease that's most researched across the world. And since the recent since the new emerging technologies over the past 10 years have improved drastically, cancer is something that is showing a big promise that we can treat. So a lot of companies are developing and researching into cancer therapies and cancer drugs. So alhamdulillah, I've worked on several different projects, supported many different projects. My day-to-day work involves going to the lab, performing designing and executing specific types of experiments using high-tech equipment. One of them is PCR, which we'll cover shortly, and then generating the data, from that data going back into the office, analyzing that data using high-tech software, and then making sense of that data. What does that data mean? How can we use that data to answer the hypothesis for that particular experiment? And then what is the next question that we need to answer? And then we present that back to the team, the whole department, so that they are also in line with the data that I've generated, so that everyone has an input. So people can say, actually, Qamar, did you think about this? Did you think about such and such experiment? Did you think about this? What did this particular result mean? And people can, there's many intelligent people that I work with who fill in the gaps of the knowledge that the gaps in my knowledge that I'm missing on my part. Other colleagues can fill in those gaps. So the two main projects that I worked on is a cancer cell therapy, which alhamdulillah has gone to early development phase. Now it's gone to practical process development, so that this phase is sort of researching into how we can scale up the product without compromising the efficacy and safety. And so once we've figured it out, it will then get an IND. An IND is a form called, it stands for an investigational new drug application. So that's the application that you submit to the FDA or the MHRA regulation bodies to be approved for clinical applications, and that's sort of the endpoint. And it's showing a lot of promise. This cancer drug. The other project I worked on is a gene editing project, which was very, very interesting, very powerful as well. And alhamdulillah, the past 5 years I've developed my skills in immunology, biochemistry, molecular biology. Now I have a broad knowledge of different areas of science that, inshallah, I hope I can use to provide perspective throughout this webinar.
So just to sort of recap for the audience, it's so you're a scientist that I sort of hypothesize things around certain treatments. You'll go through a process, a bit like how we used to do back in sort of GCSE science days, where they give you a thing to sort of work on, and you kind of work towards it, just to make it a bit easy for everyone who's listening. So that really brings us on to our I guess topic. So in your line of work,
sorry, can you hear me? The voices, can you still hear me?
Yeah,
okay,
clear.
Cool. Yes. Okay, carry
on. So just so moving forward onto our topic, for everyone who's watching, we are hoping to keep it under 45 minutes, so it won't be as long as the last one, so please bear with us. What is mRNA, and what is sort of the main thing around the vaccine and the link between mRNA?
Yeah, so I mean, mRNA, there's a lot of misinformation. There was a lot of fear mongering, a lot of scare around this new type of vaccine that incorporates mRNA into this new vaccine, and rightly so. People will have some contentions and misunderstandings about mRNA, but in fact, mRNA is present in our body. So, I mean, to really give an understanding of what mRNA is, I have to briefly talk about what DNA is. So I'll try and keep this as brief as possible. So DNA is basically, it contains all the genetic code of a human. And these genetic codes are sort of the blueprint that determine, for example, your skin color, your hair color, the color of your eyes, etc. And that's being very simplistic here. But to be more technical, DNA contains a genetic code for proteins. So proteins are what you could say are the building blocks for life, to life. So if you imagine like a box of Lego bricks per se, so the box is sort of the DNA, the Lego bricks represents the different protein building blocks, and the mRNA represents the instruction manual that comes with the Lego box. So for example, if this Lego box is a box to make a Lego house, that instruction manual will tell you how to make that Lego house using the Lego bricks in that box. So similarly within our cells, we have what you call nucleotides, and then they get joined together. So just to talk a bit about protein as well. So in 2018, scientists determined that a single cell approximately contains 42 million different types of protein within a single cell, and that's only in one cell. So just bear that number in mind. And in the human body alone, there are approximately 30 trillion cells. So as an approximation, if you multiply 42 million by 30 trillion, the numbers are astronomically large. And that's a lot of proteins that your body has to keep making on a daily basis. So every day your cells are being damaged, every day your cells are dying, and therefore new cells have to be made. So if you can imagine, that's a lot of proteins that have to be made every second of the day. So how is protein made? So within your cell, there's what we call a nucleus, and the nucleus it houses the DNA, the genetic material, and And what happens is the DNA when the cell needs to make a particular type of protein, the DNA unwinds itself. So the DNA is double-stranded. So the DNA unwinds itself, and an enzyme copies the genetic sequence that codes for a certain type of protein,
right,
on one single strand of DNA. And then once that single strand of DNA is copied, that's what is called mRNA. So mRNA is single-stranded. Once that mRNA is produced by that enzyme, the DNA winds itself back up, and the mRNA is transported out of the nucleus and into the cytoplasm of a cell. And in the cytoplasm of the cell, you have various different types of machinery in the cell, you can call it. And there's one particular type of machinery called ribosomes. And the mRNA travels to the ribosome, and the ribosome basically reads that mRNA, which codes for that particular type of protein. So it's all like a conveyor, like a production line. So if you imagine that this is a ribosome, the mRNA comes through the ribosome, and from the other end out comes a protein, and then that protein goes on to do whatever it needs to do. So if it's a protein for structure for functional or non-functional proteins, there's so many different types of proteins. So you can imagine every single second of every minute of every hour, every day of every year, your body is producing mRNA for protein production. So this is the misunderstanding that the general public have about mRNA. They think it's a new thing, it's not a new thing. We live with it every single day. It's in our bodies, being produced every second. So, scientists have figured out over the years how to synthetically make mRNA and to study to synthetically make mRNA to study and make proteins. So we study these proteins. One thing that often gets asked of me is, how with this mRNA vaccine, how long will it last in the body? So mRNA is quite labile. So labile means it's extremely sensitive to being broken down by enzymes. So in our everyday surroundings, we're surrounded by different enzymes, microscopic enzymes from organisms, and these break down mRNA. So just to give you an example, one of the first experiments I did using mRNA, the experiment didn't give me the results that I was expecting. And one of the conclusions, in fact, one of the main conclusions of my experiment was that I mishandled the mRNA because I didn't respect the mRNA. I didn't sterilize my surroundings with a particular solution that breaks down enzymes in my surrounding.
So it's very sensitive.
It's very, very sensitive. But in your cells, in your cells, you have enzymes that break down mRNA. So just to rewind a little bit, when I was explaining how proteins are made, so the mRNA comes through the ribosome and the protein comes out of the other end. Once the mRNA is served its purpose, it gets broken down by enzymes really quickly. I tried to find a specific time frame of how long mRNA lasts. It can last from seconds all the way up to minutes. It's broken down very, very, very quickly, and that's all dependent on the length of the mRNA. So
this vaccine, which is mRNA based, you're saying that it could be finished within 5 minutes, 10 minutes?
Yeah,
an hour maybe.
Once it's served, it's all to do with the dose as well. So it's all to do with how much mRNA is being injected into. Well, it's all about how much of the mRNA enters the cells. And then, let's say, to give you an example, let's say, and this is just a scenario now. Just to make it easy for the viewers to understand. So let's say we have 10 cells, and of those cells, 7 cells, the mRNA manages to get into the cell. So once that cell has processed the mRNA, which produces that protein, and in case of this vaccine, it produces the spike protein, which travels to the surface of the cell and expresses right at the outer cell membrane of the cell for immune cells to recognize and then generate an immune response. So let's say let's say it takes a minute for that process to occur. It's very, very quick. It's very, very quick. So it's all to do with how much mRNA gets into the cells and how fast the mRNA is processed. So
it's priming our immune system to respond to this via this messenger, if you want to call it.
Exactly.
This is what it's going to be like. This is what COVID is.
Yeah.
This needs to be your response.
Exactly, and that's how the immune system works. So, every day our bodies we ingest different pathogens, whether there's bacterial pathogens, viral pathogens. Our immune systems are always working constantly, and over the years we develop immunity to the most common types of pathogens that we come in contact to on a daily basis. So, but really briefly, when we do come into a contact with a new pathogen or a pathogen that we've already recognized, what normally happens is certain special types of immune cells recognize this pathogen, they ingest it, they break it down, and then they process some part of that protein on the outer surface for other special immune cells to come and recognize. So, for example, T cells. So T cells come and they say, okay, we don't recognize this protein. Yeah, it's a new pathogen. Let's go back to what's called the thymus gland. That's where T cells are processed. The T cell travels back to the thymus gland, and in the thymus gland, the thymus gland says, right, okay, this particular protein from this new pathogen that's entered your body, this protein is not present on our cells. Yeah, so you're safe to go and attack that. So that's how that cell goes back into circulation, finds that pathogen and it attaches itself and it starts attacking that pathogen. And then whilst it's attacking that pathogen, that T cell releases what's called cytokines, which causes other immune cells to come, as well as B cells, and then and it also tells the T cell to start rapidly multiplying as well. So you've got a really, really strong immune response. And so for this particular case, COVID, the protein that our immune system recognizes is the spike protein, the ACE2 receptor on COVID-19 coronavirus. So all we've done is basically we've found the genetic code for this spike protein, and we've synthesized an mRNA, and all we've done is we've injecting that back into a patient for the patient to then process this protein onto its own cell surfaces to build up immunity. So the immune cells come and recognize that synthetic spike protein, if you want to call it a synthetic spike protein, and it generates immunity basically. So next time if you come in contact with that the coronavirus, you've got B cells ready, you've got an immune response ready to at the get-go to attack.
Okay, that's wow. Okay, just on a quick side note, which I didn't ask you before, but I'm just gonna put it in now. Also, really quickly for the audience, if you've got any questions, please post it. And apologies, that is a little bit more technical than the last one. I guess some of the technicalities are quite important in the sense that you are appreciating as an audience that this isn't something that you can just forward as a text message and say mRNA is false because I've read a paper from somewhere else. So, but we will try and keep it.
Sorry, Zan. It's.
Yeah, you're fine. Yes, sorry. Slight technical issues, but carry on. We can see you. We can hear you.
Sorry, Zan. The line's gone a bit fuzzy.
It's clear here. Just bear with us, guys. We've got a slight technical issue. Qamar, are you still there?
Hello?
Qamar, are you still there?
Can you hear me?
I can see. Apologies, guys, for that. That's okay. So I was just saying that although it's technical, we'll try and keep it as layman friendly as possible without maybe going into too much detail. Just a quick side point. Obviously, you work for GSK. We just want to clarify that you haven't got a conflict of interest in this. You know, has GSK bought a vaccine now, or is any issues? Obviously, you've come on.
Yeah.
Yeah.
So I mean, GSK were in the works of generating a vaccine. They are collaborating with Sanofi. Recent results came out, I think, end of last year, that the vaccine didn't really stimulate a strong enough immune response. It wasn't as effective in elderly patients. So they decided to go back to their drawing board and trying to figure out why this has happened and then increase the effectiveness of the vaccine. So
But there's not currently one on the market, is there?
No, not currently one on the market. But I mean, GSK is a vaccines company. They have produced other vaccines in the past, and they continue to do so. But specifically for coronavirus, they're not quite there yet.
That's fine. I mean, we didn't want conflict of interest here, where people thought we brought you on to try and promote GSK in particular. No, that's fine. Okay, great. Just really quickly, if you can sum it up, what is a PCR test? So obviously, we're told that if we have any symptoms, we need to go to have this swab in our throat and up our nose. I guess what happens when that is sent to the lab? Just really quickly, if you could break it down easy for the audience, because a lot of people are saying there's a lot of false negatives or false positives or whichever way they see it.
Yeah, I mean, this is probably one of the center of the debate between in the scientific community. Is one of the debates that's causing a lot of contention, you should call it, and there's a lot of misinformation around the PCR test and the accuracy of it and the reliability of it. So, you know, I think it's important to go into slightly more depth to explain what the PCR test and to clarify some of the misinformation out there. So, you know, like I said, it's been a subject of the accuracy of the PCR debate. You know, like I said, has been a center since you can say summer last year. And again, it's really important to put things into context and perspective, because again, there's a lot of misinformation circulating around. And when the general layman reads or hears these headlines in the media or social media, they latch onto it and run with it without having any understanding of how PCR test works. So, yeah, I've performed this test several times in my career in my experiments, and yes, they are very accurate and highly sensitive. And I think the issue with the test being used for mass testing is more so, are they reliable enough? So, you know, that term, that phrase, are they accurate enough? I don't really like to use the word accuracy, because they are accurate. I think the question we need to ask is, are they reliable enough? So, but before I go into explaining how the test works, I just want to comment on some of the misinformation that's been doing around on social media by anti-vaxxers and COVID deniers. And one of the main pieces of misinformation around the PCR test is that the inventor of the PCR test said that PCR does not detect virus. And there is a quote that has been taken out of context. So the quote that they use is actually, the quote was taken from an article written by a chap called John Lauriston, not actually from Kary Mullis himself. John Lauriston, he wrote an article about protease inhibitor drugs for the diagnosis of HIV and AIDS in gay men. So this was the context of this is the context of why PCR was mentioned in his article, was to detect HIV virus in gay men, and he referenced Kary Mullis and the PCR test. So I read this article, and what struck me was that in the article it clearly states, and I'm going to quote this: the tests can detect the genetic sequences of viruses, but not viruses themselves. What PCR does is to select a genetic sequence and then amplify it enormously. It can accomplish the equivalent of finding a needle in a haystack. and it kind of amplified that needle into a haystack. So what was said in that article is the truth. It doesn't detect the virus, it detects genetic sequences of the virus. And this is the biggest misconception that the general layman has about the PCR test. And the statement is true. However, this was taken out of context again by anti-vaxxers and COVID deniers. But you know, the PCR is a test to select genetic sequences in DNA or RNA of a cellular organism, whether that's cellular organism, a bacterial virus, or a human cell, but not the actual whole viruses itself. And in that article, it specifically says that. So I'm going to go into briefly explain how the PCR detects these genetic sequences in layman's terms. So if you, so the PCR test, you can think of it as a control F function on your Word document. So
okay.
So let's say if you have a Word document that is, I don't know, 100 pages long, and you want to find the phrase, I don't know, just you want to find a specific phrase, in that Word document, rather than having to read that whole 100 pages before you find that document. And so what the software does in that, what you do is you input the phrase, and the software then highlights every single section of the document that contains the particular word or phrase that you put into the search box, and ignores everything else in your Word document. And this fundamentally what PCR does. Similarly, in the test in principle does the same thing. So you design the PCR test to only find a specific genetic code, ignoring all of the rest of the genetic code present in the sample. So it will ignore human DNA, it will ignore bacterial DNA, and it will only find the genetic sequence of the viral mRNA that you're trying to locate. So when you perform PCR, so a swab is taken from the patient's mouth and the nose, and on that swab contains cellular material. So when you do the swab, you have cells that get stuck to the cotton bud, and then you put it into a solution, and the solution acts like a buffer. So by the time it reaches the lab, a lot of the cellular material has now leached into that solution. Because in science we work with solutions, we don't work with cotton bud. We then take that solution, and that solution is then processed using a range of different chemical or biological reagents. And what we want to do is break down all of the cells in the sample so that we can access the DNA or mRNA contained within the cell and in the cell nucleus. So all of the broken down cellular debris and other contaminants are washed away and filtered out, leaving only intact DNA or mRNA. And then you add what is known as primers, and these are called DNA primers. So the easy way to explain what DNA primers are is you can think of these as like magnets or anchors. So let's say my finger
extracting it out.
The prime is attached, basically. It's like an anchor. It's like a magnet. So let's say my finger represents the genetic sequence. So what you want to do, you got to imagine in the sample you've got loads and loads of DNA or mRNA. So how are we going to find that specific genetic sequence that matches the coronavirus? So we know the code, we know the genetic sequence of coronavirus, and so we attach primers on either end of that sequence. It acts like an anchor. So once it's attached, the machine then starts to make mRNA into DNA. So it does this by reverse transcription using reverse transcriptase enzymes. So similarly, how I previously how I mentioned how DNA and mRNA is made from DNA. It's a similar process here. You have to convert the mRNA back into DNA, and then that DNA is replicated. So along with primers, you add a fluorescent probe. So what that fluorescent probe does is attaches to the genetic sequence of the coronavirus within your sample. So what happens is when the machine, when that genetic code attaches it to itself, it lights up, it gives a signal to the machine, and that machine tells you, right, okay, we have coronavirus present. But I have to speak a bit more in depth about the amplification cycle. So in PCR, we have what's known as amplification cycles, right? Yeah. And usually in a normal in a research setting, PCR tests usually go for 30 amplification cycles. And the reason why we have so many cycles is that if there's only a tiny amount of genetic code that you're looking for, it's like, again, like I said before, it's like finding a needle in a haystack. So by making more and more copies of that genetic code, by amplifying it, you know that after 30 cycles, if there is a signal above a certain standard threshold, you can accurately say that the genetic code is present in the sample pertaining to that virus. And normally this test is quite accurate. It's very sensitive, and within a standard research setting, it has between 1% to 3% error. So the error is very, very, very small. So what the specific thing that came up last summer was that a few scientists across the globe came out towards June, July, and said, hang on a minute, what the PCR may have some problems, and what we might think is positives, we actually picking up false positives from the number of cycles being used to detect COVID-19. And
what does false positive mean?
So false positive means basically if you've a sample that shows a positive when in fact it's actually negative. So
okay,
yeah. So a negative a sample tests positive, but is in actual fact negative. So that is what a false positive is. But I mean, just going back to what I was saying, so the standard number of cycles that we conventionally use with PCR is 30 cycles. And this means that any genetic material that is detected past 30 cycles is highly likely to give you the false positive. So this is 30 cycles is a standard set of cycles that is generally used in PCR around the world in research labs and research settings, and this is common knowledge in amongst the research research community. 30 cycles is sort of the optimum number of cycles. You know that if a signal is picked up anywhere along those 30 cycles, it's going to be positive and accurate. But
so was our government doing more than 30 initially? If that is the case, playing a bit of devil's advocate, so could initially, when the pandemic hit, could there have been a lot of false positive initially?
Yeah. I mean, it's a valid argument. I've looked into both pros and cons of this, and it is a valid argument. But in my humble opinion, I personally believe that. I mean, look, it was determined that the government were using 35 to 40 cycles. And this was the key point here, that the scientists came out and said, you're using 35 to 40 cycles. This is not reliable. It's not reliable. You're picking up a lot more false positives than normal. But in my humble opinion, I personally believe that the reason why the government adopted those 35 to 40 cycles is because that at the start of the pandemic, there was very little science known around COVID-19. Nobody knew the proper mode of transmission. It was chaos, as we saw Wuhan and Italy and Spain, and then the UK. Everything happens at such a fast rate, and the UK was, poorly prepared, as with the rest of the globe. So by having more than 30 cycles in the PCR test, what you're then doing is you're leaving absolutely no room for error. Now, let me just explain what I mean by that. So if an individual has low levels of virus present and is asymptomatic, by declaring them as a positive, you're mitigating the risk of spread of COVID-19 through asymptomatic people. Now, COVID-19 in the literature, it's being predominantly spread by asymptomatic people. You also have to note that the virus has an incubation period as well, between 5 to 6 days before any symptoms appear. So this means that from day 1 to day 5, a person who has COVID-19 and isn't presenting any symptoms will be spreading this virus to everyone he's in contact with for 4 days before he knows he has the symptom. So I personally believe that increasing the number of cycles to capture any potential infectious people was the right thing to do, in my opinion. And again, these scientists that came out are fringe. They're on the fringe. I mean, I'm not going to discredit their knowledge, because these are very, very knowledgeable scientists. You know, there was a professor at Oxford University. You know, there's a handful of scientists, but the majority consensus do agree with the PCR. Now, I just want to explain this in layman terms now, why 35 to 40 cycles was adopted. So you could think of it like a fishing net. So let's say you go fishing and you want to catch as many codfish as possible, right? Yeah. This is just to make the viewer understand. So you could think of PCR as like a fishing net. So let's say you go fishing and you want to catch as many codfish, but in the sea you've got many different species of fish. So the larger your fishing net, the more cod you're going to catch. So, however, if you reduced the size of the holes of the fishing net, you're going to catch more cod. But what that also does is you're going to catch more of other types of unwanted fish that you don't want to catch. But the overall yield of your cod is going to go up, because you're reducing the number of holes, the size of your holes in the net, and you're increasing the size of the net. Similarly, the more people you test, the more infected people you're going to pick up. But by increasing the number of cycles, so the analogy is relevant for this is reducing the size of the holes of your net, you're going to catch more infected people. However, you will also catch people who aren't necessarily infected and showing up as false positives. So therefore, the overall percentage of false positives will increase. But okay, so what if the false positives increase? Ultimately, what you're doing is you're going to minimize the spread of virus, and ultimately this will lead to the reduction in the numbers of deaths by self-isolating upon a positive result. So
because
that makes sense.
The government didn't initially know how to deal with it. Therefore, that's why those cycles were selected. Okay, no, that's fine.
Yeah.
Just conscious that we have around. Sorry, I'm gonna have to cut it slightly. I'm conscious we have about 20 minutes. So just for the audience that are watching, if you have any direct questions, please drop them in the comments box. We're just gonna have to move forward a little bit quicker. So one of the big things was, what does it mean by red tape is lifted? You know, how did they get through the safety issues so quickly? You know, or has this research always been around, but it's been sped up in the last year? You know, just maybe 2-3 minutes on that.
Yeah. So again, there's a lot of concerns about in the general public of this vaccine is produced so quickly, and yeah, it's okay to have these concerns, you know.
Yeah.
Because we know that drugs typically tend to take 10 to 15 years before they hit the market. Sorry. And we have to understand that we're in a global crisis and a global emergency, and there's a deadly virus spreading across the globe, highly infectious. It's causing debilitating symptoms, and the body count is increasing. The whole economy has come down. People are losing jobs. Mental health is declining. The whole country, not just the whole country, but majority of countries across the globe are in crisis at the moment. So the only way the scientists came out, the big pharma came out and said, they put their hand up and says, we will be able to help. The only way we can quickly generate immunity to this virus is through vaccine. Yeah. But the government basically said, okay, how fast can you get this vaccine to us? You know, big pharma said 2 years, because the mRNA vaccine and also the AstraZeneca vaccine, they've been carrying out preclinical research for many years before this. So it's not necessarily new. It's been around for a long time, you know, and they've been doing a lot of tests, they've done a lot of animal studies. It's ready to go. It's at the stage just before clinical trials. So, you know, companies like AstraZeneca, Oxford Uni, Pfizer came out and said, we've got something ready. We can get it ready and And the government said, okay, can you reduce this? We don't have 2 years. It's logistically, it's going to be a logistical nightmare, and it's going to cause more problems if we prolong this lockdown for 2 years. So the government said, what do you need? Farmers said, look, we don't have the manpower, basically, yeah, because experiments take a long time. A typical experiment of what I do in a lab takes a month, from planning to execution to data analysis, and then, understanding the results. It takes typically 1 month to perform an experiment. So how are we going to speed this up? So the only way that we can do this is through financial backing. So, and these big pharma companies only have, yes, in the grand scheme of things, to us, it's a lot of money, but when you're working in a big organization, when they're churning through money, a lot of, billions of pounds are being invested. When you're looking at that ratio of money, it's not a lot of money, at that large scale, because experiments cost, investments cost, the experiments cost. You've got cost of goods, you've got to pay the workers, you've got to pay the salary, you've got to invest in so many different things. So the government said, look, right, don't worry about money, we'll give you the money, just get it done. But the and the government said, can you get it done? Can you get us a vaccine that is safe and effective? These companies said yes. So, but the companies turned around and said, okay, what you have to understand is that we won't be able to do the long-term studies. The long-term studies will have to be ongoing as we roll out these vaccines. So the government said, don't worry about it. The companies also said, okay, so you have to give us indemnity. You have to give us indemnity so that we don't get sued, because you can imagine it's emergency, people are dying, in the age of, we've seen from, since March 2020 up until now, that the rise and surge of misinformation, people will inevitably start suing these companies. And especially when the long-term studies haven't been done, people will jump on that bandwagon and start filing, vaccine injury claims against the company, and companies won't be able to sustain that. So the government said, don't worry about these people. Yeah. If there are any vaccine injury claims, we will take care of them. We've got a scheme called the Vaccine Injury Compensation Scheme. Yeah. You won't be liable for any of this because it's an emergency. So the government.
So just want to clarify that point. Really bring it home. That yes, there's indemnity with these companies, which people are saying that the way it was put forward was, well, if something happens to me or my child or whatever, they aren't liable. But actually, there is a scheme there.
Yes. Yeah.
It's a bit like a car accident, whereas if you're not taking money from the person in the car, it's actually the insurance company that's dealing with it. So I don't know if that's the right analogy, but it's the same thing. So there is money available if something went wrong. Big if.
Yes, that is
the company.
The analogy of a car insurance is exactly what it is. So, for example, God ever protect me from this, but if I was going to go out and I accidentally run over someone, caused a serious accident, that was my fault, and it was clearly an accident. It wasn't intentionally done, and I won't be able to personally compensate the loss of that life, but my insurance company will. Does that make sense? So
yeah,
because I don't have that type of money, I don't have that type of finance and that capital, and that's not to say, and that's not necessarily a bad thing. It's to protect these companies from exposing them from this type of risk. Because if that was to happen, then what will happen is you got a whole pile of cases coming up, right? Yeah, you can bankrupt big pharma like this. Big pharma can be in real deep trouble, and what will happen then is the life-saving drugs and treatments that they're already producing, that's going to be put at risk. So, for example, let's say GSK, they produce salbutamol, an inhaler for asthma. Let's say, as a scenario, it happened to GSK to produce the vaccine, and vaccine injuries started cropping up, and then claims were made against them, and they had to pay out large sums of money to lots and lots of people, especially during this pandemic where the whole world is being vaccinated. Yeah, what will happen is you're putting their current drugs that are already treating patients for certain diseases, like for example, asthma. I take a blue inhaler. You know, what will happen is that I won't, if the company goes under, where am I going to get my inhaler from? So these
So it makes
these
sense.
companies are doing good work. They're making life-saving drugs. They're making life-saving treatments for different types of
And
diseases.
just on I guess a personal note or question to you is, yourself, your colleagues, your managers, your supervisors. Obviously, people will have this thing about big pharma, these companies. It's about money, 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 money. But you must genuinely want to help people out there, whether it's with the COVID, whether it's with, for example, in your area of cancer.
Yeah,
There's going to be a genuine want to help through this, whether it's a vaccine or a drug or whatever it is. Is that correct?
of course. Yeah, of course. You have to. Yeah, of course. You know, drugs aren't cheap. You know, there's drugs in an average drug costs 2.1 billion. I think one article 2.1 billion. You know, let's break that down. That's 200. That's $2,000 million. Let's turn it into, that's 2 billion is 2,000 million, isn't it? Yeah, it's a lot of money. You know, when a drug company invest in a drug and it shows safety and efficacy at the preclinical trials, at the early development phase, and then it's ready to be approved for clinical trials. The company is already minus £1 billion. You know, they're already in negative £1 billion. It's a lot of money to not make returns on. Fundamentally, it is a business, and there's nothing inherently wrong in a business. You know, if you strip away everything and just of a business and look at what business is, a business is, you buy a product or you make a product and you sell it for profit, and then from that profit you reinvest it back into the business, into new drugs, into new parts of your business. You know, you pay your workers. And just to give you an idea, or just to give an idea of the viewers of how expensive drug development is. So, and this is the naivety I came when I first joined GSK. I have to admit, I was one of those COVID. I mean, not COVID, but I was one of these conspiracy theorists. You know, I did believe in some of them, and one of them was drug companies. I thought, yeah, drug companies only in it to make money from people who have diseases. And when I joined, and you know, over the course of 5 years, it's all of those conspiracy theories left my mind, because I saw what's happening. I was involved in developing drugs, and just to give an example, a typical experiment can cost anywhere between £500 to £5,000. That's just for one experiment, and a lot of the times the experiments don't work. And you have to repeat your experiments, and those experiments need to be repeated 3-4 times. And then that's just one individual. You know, in GSK, where I work in Stevenage, there's over 2,000 workers. And let's say, for example, a department has 50. A single department has 50 scientists. Yeah, of those scientists, 25 are in the lab performing experiments, and 25 people times 5,000. What's that? You do the maths. It's a lot of money. And that's just for one experiment. And then you multiply that across the whole globe. Drugs aren't cheap. Drugs cost a lot of money. And yeah.
I'm going to have to just move forward. We've got about 10 minutes, but we still had a lot of questions. So I'm going to have to cut some of the questions out that were sent in and just try and go through the quite important ones. Around the vaccine, so the manufacturing of it, there's been talk about the government giving, for example, dose 1 as Pfizer, and experimenting the second dose as one of the other ones. Now, some epidemiologists have, at the moment, strongly suggested they shouldn't be mixed. Is this something from your background in research that should be done? Is it experimental, or do you think it might work, depending on tests?
So I've looked in the literature and to find out about mixing of vaccine, and I mean, the general guideline here is that they shouldn't be mixed, unless on a very rare occasion that vaccine, that's the first dose of vaccine, is not available for a particular individual who needs the second dose. So let's say an individual needs a second dose, but it's not available, and that's the only scenario where they can mix vaccines. But in general, in this COVID-19 vaccine rollout, the guidelines are clear, they shouldn't be done. But there is literature on other vaccines that is showing promising results of mixing vaccines that contain that target the same antigen, basically. And in some papers, it actually shows increased immunity. And so this is ongoing research, amongst epidemiologists, the immunologists. Yeah, but
okay, and obviously on the actual topic of COVID, the actual issue, if there's a huge antigenic drift, so if it mutates to a point where it's almost different from where it was at point one when it first came around, we're aware that there's a lot of variants, but a lot of the vaccines seem to still be effective against majority of the variants. What if there's a huge change? I mean, will the companies be able to come out with a quick, booster vaccine within 3 months? Could we be stuck in a cycle where it keeps changing every 3 months because not enough people are vaccinated?
So if that huge antigenic drift was to occur, then theoretically speaking, the current vaccines will become void. Yeah. And we would need to produce new vaccines to the new strain of virus. But with these current new technologies, so the mRNA vaccine, it's easy to tweak. The science has been done on it. The clinical trials have been done on it, and mRNA, as I explained, at the beginning of this webinar, is very easy to synthetically make it and tweak the genetic code for that particular strain of virus. And what I've read is that they won't need to go through the whole clinical trials again. They will still need to perform a small clinical trial just to show that it's safe and it's effective, that it's effective, and show the level of effectiveness it has towards a new strain. But this is the beauty of the mRNA vaccine, and also the Oxford vaccine as well.
Okay, so the blueprint's already there. In short, so I assume the way the mutation will happen is it's always the same type of sort of the drift. Although it'll be different, it will still be the same type. Therefore, the vaccine can just follow the switch again and again and again, if needed, to a point hopefully where it fizzles out.
Exactly. Yeah. Exactly. The framework's already there. The production plant's already there. Everything's there.
Okay. So we've heard some doctors talk about and specialists say these pandemic style viruses can happen again. So there's this big thing about Bill Gates saying back in 2015 that there could be a pandemic coming around the corner. So unless he had some secret knowledge about it, is this well known within the world of science? And could there be things coming out in the future?
Yes. So these are things that countries try to prepare against, and especially epidemiologists and virologists. They study, viruses, and they perform, calculations. They basically do the maths and basically try to estimate, okay, when will the next pandemic occur? We saw the last one. What was it? The Spanish flu, and obviously they studied pandemics even before that. So they knew something was coming, but they didn't obviously know it would come in 2020. But they have things in place to. I wouldn't say they have things in place, but they try to anticipate when the next pandemic comes, and they're always studying viruses, especially for coronaviruses. Coronaviruses are highly studied because coronaviruses are present in the common cold.
Okay. The vaccine as an actual exit strategy, just quick 20 seconds on that. Is it the best exit strategy we have, or is there other alternatives?
Well, it's the only exit. It's the only exit strategy you have. It's either immune the population or let the virus rip through the population, and your body count's going to go up. You take your pick. It's as simple as that.
And obviously, the preservation of life is
exactly
is very, especially from our Islamic perspective. If we leave the just the pure science aspect, preservation of life in the Sharia is very, very important. So
exactly,
that's all the way forward. Again, just a quick one. What happened to the flu?
Oh, this is the question that I asked quite a lot. The flu is still there. The flu is still there. So just really, really briefly, flu season happened, peaks between December and March, right? Yeah. So you think about that, the coronavirus came and lockdown happened. The first lockdown happened, on the 23rd of March of 2020, I think it was. So technically speaking, the peak of the flu season is dying out now. Yeah, everyone's had flu. The flu's peak is coming down. But when this happened, everyone went into lockdown. Everyone's in their homes. Medical guidelines such as increasing our hygiene standards, so washing your hands, using alcohol rub, social distancing. These all, contributed to slowing the transmission of flu. So by the time winter came back around again, so by the time September, the autumn and winter season came back again, everyone was still washing their hands. When we go to the supermarkets or shops, people were still observing social distancing, people wearing masks. So the flu didn't have traction to pick up and spread again. But COVID-19 was already in the population spreading, and it's more of an infectious disease. But flu, we have seen the lowest flu rates ever in the past, I think it's 100 years, I think I've read.
But the flu, I guess,
yeah,
it makes sense. If you think about it, from a logistic point, it's you're regularly washing your hands, you've got a mask on, you're staying away from people. You don't actually have an opportunity to catch it. Exactly.
Once everything goes back to normal, once the social distancing ends, lockdown ends, once the mandating a mask is no longer, the flu will pick up.
So you mentioned two things in that point, and I'm going to put these two questions together. Obviously, there was a study by Zishan Qureshi and Nicola Jones and others who spoke about the 2-meter rule doesn't really apply, or shouldn't really be applying, because it's on an outdated dichotomy model. They sort of suggest about an 8-meter area. What do you think about that? Is the 2-meter rule still valid? Is it still worth doing?
Yeah, the 2-meter rule is still worth doing. What you got to realize is you have to incorporate practicality. So if you think about 8 meters, if you were to go to a supermarket and everyone had to social distance 8 meters, nobody A is going to adhere to it. B is practically it's a practical and a logistical nightmare, and it's not practical. That's the best answer I can give. It's completely impractical. 2 meters is sufficient, especially when you're wearing masks as well, and especially for outdoor situations when you're outside. When you're outside and you're wearing a mask and social distancing, that significantly reduces the transmission of virus particles via airborne droplets.
Yeah. We just got a question coming through the Google form. I'll ask it if you feel like you can answer it. So the question is, why is there heavy focus on the questionable germ theory when terrain theory counts for so much more? Everyone has a responsibility for their own body and what they put in it and how much they look after it is an amana. Why inject it with a toxic cocktail of chemicals? I guess we're talking directly about vaccines and things that are put in.
So I've heard of the terrain theory and I've heard of the germ theory, and I have to hold my hands up and say my knowledge is very limited on this. And again, people have their own choice, free will to take whatever they want, and nobody's forcing. The country is not forcing. Nobody's forcing you to take this vaccine. What you got to remember is as well, we're taking and ingesting chemicals on a daily basis, whether that's through our food, for numbers, whether we're using our multivitamins, general medications such as paracetamol, ibuprofen. These are all chemicals that we ingest and we don't even think about. So vaccine, okay, there's a lot of concerns around it, but again, it's a drug. At the end of the day, it follows the same generic drug process, the same drug development guidelines, go through the same approvals, and vaccines are probably one of the most studied and researched drugs, in the science industry. But
yeah, I mean,
that's all I can comment.
At the end of our, before we finish, our last question is going to be on sort of a bit of vaccine hesitancy, which people want to know. But before we get there, so the COVID vaccine is obviously not going to prevent people from catching COVID. It's about a response, right? As you mentioned earlier, just to maybe recap quickly, it is an immune response.
Yeah. So when you take the vaccine, it won't stop you from catching COVID. what all a vaccine does is it's training your immune system to recognize the virus, and it primes your immune system to generate antibodies, so that they're ready at the get-go in case that virus or pathogen comes into your body. It's a bit like, how can I put it now? It's like a bit like you get mugged on the street, and you've got no martial art skills, but you recognize those faces, and so you go to the gym, you go to the martial arts gym, you train up, you become a superstar MMA fighter. You see the same people on the street, but this time you're prepared, you're ready to to defend yourself. It's a similar analogy to a vaccine.
This question, can't spend too long on it because I know we had a conversation pre-hand about this. So a lot of people thought that through the vaccine, DNA would be edited. Now the doctors, in short, had said no, it won't edit the DNA because it doesn't actually go in there. Is there DNA editing at all that's happening at the moment? Is it an issue, or is there anything to do with vaccines?
So I mean, I can answer this quite well. So I've done a gene editing project, and the type of drugs that I've been working on, the cancer drug, it involves editing of genes. But in this particular vaccine, there's zero gene editing at all. The vaccine will not, and I categorically say will not edit your own genes. It's just it's just not how it works. mRNA will not get incorporated into your DNA. A lot of things, the way the mRNA is designed specifically not to do this. It's designed specifically not to enter your nucleus. Once the mRNA, your own natural body mRNA is produced, it leaves the nucleus. So if you're putting mRNA into a person and it enters the cell, it's not going to go to the nucleus. It's just going to go straight to the protein factory in your cells to produce that protein. It would not at all genetically modify or incorporate into your own DNA. It's just laws of biology and protein chemistry, biochemistry,
and it seems like a lot of doctors and specialists are coming out now saying that these vaccines are in fact very safe, and I guess they're getting to see the amount of people that have been vaccinated. That there's been, I understand there shouldn't have been any deaths currently when it came out. I think
30 million people have been vaccinated. I think it's the Oxford vaccine, or one of the vaccines. 30 million people have been. I think it's the total amount of people that have been vaccinated. There's not been one death associated with the vaccine. There have been deaths. I mean, I'm not going to say there have been deaths, but these deaths aren't linked to the vaccine. Also you got to remember is timing as well. The timing of these vaccines, as well as the coronavirus still present in the population.
Coming towards the end, this is quite important, I guess, and it's worth touching upon. So this vaccine hesitancy or anti-vax movement and things like that. I just think it's worth touching upon two of the things. The first one, if you can, on the correlation between vaccine and autism. So what happened in that period and how it started. And just the final one, just to show people that you can have a lot of things out there that come up about, this happened with the vaccine, but there's always like an answer, or at least which was about the COVID-19. But we'll start with this one first, the autism one.
I mean, so over the past year, I've been quite active in debunking a lot of the misinformation, because it in my opinion, this information is even more deadly than the virus. It's causing. I know people who have bought into this misinformation and are now at a loss. Whether their loved ones, some of the loved ones are maybe in hospital, some of the loved ones have passed away. It's really, really damaging, the misinformation that's out there. But what people don't understand is that there is a source. If you go back to the source, you look at the history of where vaccine hesitancy first came around. Vaccine hesitancy has been around for over 100 years, But in our modern time, the anti-vax movement started to pick up around about 1998. And what happened during 1998? There was a British medical doctor by the name of Andrew Wakefield. Now he had this hypothesis that the MMR vaccine, the measles, mumps, rubella vaccine, causes autism. He said that there's a link between them. So he performed a study on, I think it's 12 children. So automatically, the number of patients using it is very, very small. It's not enough to come out with a conclusion on such a small cohort of children. So as he did this, he performed unethical medical procedures on these children. He also had a lot of conflict of interest. So he was trying to show that he was trying to smear the MMR vaccine, which is a trivalent vaccine. So it vaccinates against rubella, measles, mumps, and rubella. Yeah, and he wanted to show that a single vaccine is better. Now, it came out over the course of 10 years that he had a conflict of interest because he filed a patent for a vaccine that he was trying to generate himself, which happened to be a single vaccine. Also, he had the financial backing of a lawyer who was supporting parents who had children a lawyer who was financially backing him, who was supporting cases of vaccine injuries. So there you go. That was another conflict of interest. But one of the biggest things that what happened was this paper was published in the Lancet, and the Lancet is a very well-known, highly reputable scientific journal. And normally, when you're looking at when you're researching papers, one of the key things of one of the things you get to know when you're in the line of research is where to pick information from. You know that if you read a medical or scientific article from Nature or the Lancet, you know it's going to be quite good, yeah, because they're quite stringent. They peer review their papers, but every now and then, unfortunately, one paper always slips through the net, and this happened to be one that slipped through the net, and it remained in the Lancet journal for 10 years. So over the course of 10 years, a British journal started to expose these fallacies, misconduct, gross medical misconduct, conflict of interest, and also the science was, he couldn't reproduce the science. His conclusion did not support his claim. So over the course of 10 years, there were about 6 independent studies that actually wanted to see if they could reproduce his research and to see if there is an actual link between autism and the MMR vaccine. And in total, in combination, these 6 studies looked into over 150,000 children, and not one of those papers found a causal link between MMR vaccine and autism. It was just not there. They couldn't. The findings were quite robust. So by I think it was 2010, his paper was taken off from the out of the Lancet. He was stripped of his medical title. The 10 authors that in total that paper had 13 authors. 10 of those authors retracted themselves from the interpretation of the findings of those papers. So he basically fooled his co-authors into performing this research with him. Also, the children that he conducted the research on, their medical records, he falsified a lot of his results and falsified a lot of his medical records in that paper. So there's a lot of gross misconduct that happened. But what happened was, this over those 10 years, and the anti-vaccine, the damage was done by 99. When that paper came out, the damage was done. Anti-vax people jumped onto it, said, here's a reputable. They fell into the trap, the biggest trap of believing into these theories is called appealing to authority fallacy. Now, basically what that means is if someone has a doctor's title or PhD or is a professor, the general layman generally trusts these people, because it's like me going to a doctor. It's like me going to the doctor's surgery and ask him and just let him know that I've got a certain condition that I want him to look into. I wouldn't question my GP because I have trust in him. I know that he studied over, many years of his life. He's an expert in what he does. And this is what happened. People fell into that trap because he was a medical doctor. And he himself started to push anti-vaccine propaganda. And he is still doing it to this day with the COVID-19 vaccines. And he's, over the years, he's gained a mass gathering of anti-vaxxers. Now we've got a huge movement, and the damaging reputation vaccines have, I believe, started from this point onwards. So what the key point here is the key point here is that research is something that you can't just do by looking on Google. You have to know what you're looking for. Research, you have to eliminate every single type of bias, whether that's confirmation bias, personal, emotional bias. You have to look at the reputation of the journal that they come from. You have to look at the authors. You have to actually look at the methods that they use in the research paper, because there might be a method that they use, but in actual fact, there's a better method that they did not use. So which makes their method in that particular paper redundant, and makes that paper weak. It's a bit like hadith. It's like hadith. So you got Sahih, Hassan, and Daif. Yeah. Now you take the Sahih because it's 100% authentic. Yeah. But you leave that you take away the weak Daif. You don't really take from the weak Daif hadith, because there's discrepancy. There's discrepancy within the chain of narrators. There might be a narrator in the chain that might be a liar, or who has bad memory, that goes on to make this hadith weak. And it's a similar concept in research. You have to really verify what your information comes from.
So with him, basically, the damage was done. The movement had started. Because even when I was looking into vaccines, I kept remembering in the back of my head that, do these things call vaccines? It was a genuine question, because it's just pushed around, and you don't really know where to, how to even have a look at the research.
Yeah, one thing that tends to happen is these guys get labeled the unsung heroes. I mean, like whistleblowers. So in modern times, like Julian Assange and Edward Snowden, how they expose a lot of government, fallacies, and they did good work. They exposed a lot of these fallacies, but and they've been labeled heroes, haven't they? The people view them as the unsung heroes of our times. And unfortunately, this, in terms of the anti-vaccine movement, there's unfortunately a lot of bad apples in science, in the medical profession. You can get bad people from any profession. The biggest example I give is Harold Shipman, the serial killer. Just because he did what he did doesn't mean to say that every medical doctor is bad. Like similar with police, you get bad police officers, as we saw last year with the whole BLM movement. But does that mean that we don't need police at all? We do need police to police our streets and keep us safe. And similarly with research, we do get bad scientists who perform bad research and publish bad research, and it gets then misinterpreted incorrectly by the general layman.
Okay. That's really good. I hope people really uptook that. So we'll come to the end of our webinar today. If you could sum up what you want people to take away in about 60 seconds, what was key and how they can move forward.
I think the important key things are really verify your information. If you don't know, or if you have a doubt, what you always got to understand is there's always two sides to the story. If you have this mentality in mind and you read a piece of information, try and find the opposing view of that, and then make the judgment. And if you don't know, take it to an expert. Vaccines are completely safe. The large body of scientists around the world have come to a general consensus that they are safe. And scientists, doctors, we all have the best interest of our public at heart. So really go to them and put your trust in them, and don't take anything from social media. If you have any doubt, take it to the experts.
Okay, Jazakallah khair. That is Qamar Hussain. If you have any questions around what's been spoken about today, please feel free to message Islam Bedford. We can pass it over to him if there's anything more in detail that you can check. We hope you enjoyed this webinar. Just again, as another disclaimer, we aren't, as Islam Bedford, saying yes, take the vaccine, or no, don't take the vaccine. It's purely from a informational point of view. And today's webinar does not hold any medical advice. So please don't come here and say, oh, you know, the senior scientist has said everything's safe, I'm going to get it. Please obviously speak to your local GP and things like that. Please subscribe to Islam Bedford's Twitter and Facebook. We're planning on doing some more webinars like this coming up in the future. So we're looking at planning out one for spirituality around COVID and what happens when you pass away. There'll be some educational ones for things like what careers can we take forward for people between say 14 and 18 and things like that. So please stay tuned. Also in Bedford, there's 3 vaccination centres. They are Bedford Heights, Manton Lane, Sikh Temple in Queen's Park, Fordham Road, and King's House on Amtel Road. So if you need any details around that, please check the NHS website or call your local GP surgery. Obviously, it's been a very difficult few weeks, few months, even for the Muslim community in particular, coming from our, well, we're called the BAME group and background. We've had a very difficult time. Even personally, I've seen a lot of messages, particularly from the Pakistani and Bangladeshi community, of a large amount of deaths. So please remember to stay safe, keep apart, follow the government guidelines, keep on top of your masks and your hand hygiene. You know, all those who have passed away, you know, may Allah accept them as shaheeds and give ease to all of the families. And we're still going through this crisis, so please don't think that the vaccines are here and that's it. It will be finished by this summer. It will still take quite a long while. So yeah, take care. Make dua for everyone's safety, and jazakallah khair for listening. Assalamu alaikum wa rahmatullah.
Assalamu alaikum.